flexible ligand docking, or if you are keeping both the receptor and the ligand, then you call it as flexible docking. Okay, so both receptor and ligand you are keeping it rigid. Nowadays we are not using this much. Okay, so it is very fast. It's very good for high throughput screening. When you have large database of ligands, you can use this particular type of docking, but this is very less accurate. Okay, so when you allow the ligand flexible but you are keeping the receptor rigid, okay, you are calling this as rigid receptor and flexible ligand docking. So it is widely used. It allows the ligand to rotate or flexible. So computationally you can do extensive search and is more accurate. So we are going to use or implement this particular method in our study. Okay, so again, another form of docking is when you are keeping the receptor also flexible and ligand also flexible. So two ways you can do it. So you are only allowing the side chains of the some selected residues. Selected residues are those which are important for interaction or binding at the active site pocket, right? So those residues you can select and make the side chains of those residues flexible, okay, as well as the ligand to be flexible. So this is time consuming. Okay, so when you do virtual screening, we try to avoid this kind of screening. Okay, this method. Again, if you are looking for I mean more flexibility, okay, you can make the backbone atoms of the receptor also flexible, and when you are making the backbone atoms of the receptor flexible and also the ligand, then it is called as the induced fit docking. Again, this also it's for some few molecule you can do screening. Otherwise, if you are looking for 10,000 molecule screening, this induced fit docking is very it consumes uh, more computational power. Okay, so it's time consuming. So when you are sure that some of the molecules can be uh, you know particularly active against the particular target, then you can use this induced fit docking, or otherwise if you want to see the changes in the active site pocket. So when you say induced fit docking, when you are allowing the backbone atoms to be flexible, you you can generate different conformers. Okay, so when you generate different conformer, it will also change the volume of the active site pocket. So you can see if this particular changes or conformation of the receptor molecule of the active site can give you more better binding affinity or not for a particular list of molecules. Okay, based on that we can screen it out and see even when you are doing with the only single conformer, this particular list of molecule may not be having some kind of good affinity, or you may not be able to find molecule which can give you proper binding affinity. But when you do induce fit docking, the the active site pocket will be changing. So when they change, before it was not able to fit inside the active site pocket, but when you do induced fit docking, it may be fit inside the pocket and may give you a better binding affinity. And maybe this can be the uh, experimentally this can be also the active site high binding affinity molecule. Okay, so in in those kind of studies you can do induced fit docking. So again, we can categorize docking into blind docking and targeted docking based on the binding site, right? So when you don't know the active site pocket, okay, or when you don't have the information about the active site pocket, you can do blind docking. Okay, so you have this target molecule or the drug target, can be protein or enzyme, right? So you don't know where the active site pocket is. So now what you can do is you can tell the software or the program, docking program, that let me just see where this particular molecule can fit. Okay, so now you are telling the software or the program that try to see where it can fit this particular drug molecule or potential drug molecule. So it will look for everywhere. So you can define an enclosure telling that I don't know the active site pocket, so take the whole protein and do the docking. Okay, so when you do like this, it is computationally more demanding and also error prone, but sometimes it can give you a potential binding site, right? So when you say targeted docking, you know the active site pocket. Okay, so in our case also, in the example that I given before, so let's say this is where the active site pocket is. That means I already know the active site pocket. Right, so now you are telling the program that here is my active site pocket, so do the docking or fitting of the molecules or the potential drug molecule at this region only. Okay, don't look for fitting outside this particular enclosure. So when you say like this, or when you want to do docking like this, it is called targeted docking. So you are defining the active site pocket and the program is only looking for docking, or only doing the docking at this particular enclosure. Okay, so this is faster because it's not going to look anywhere else but here only in the enclosure, or in the predefined box, and you can do extensive search and it is more accurate, right? So we are going to do this targeted docking as well as blind docking. Blind docking, we are not going to take the whole protein because it doesn't make sense. And what we are going to do is we are going to try to predict the binding site, where can be the binding site or active site pocket, using various tools, and then evaluate this particular uh, docking results with some reference or uh, reports that are already known. Okay, so let's see how docking proceeds. Okay, I will just read and try to explain again. So ligands are allowed to fit at all possible positions on the ligand binding site with all possible conformations. Many search algorithms are available to perform the task. So let me just try to explain this. Okay, let's say we have a molecule here. Okay, we just got a molecule here, small molecule that we are going to see